Bismillahirrahmanirrahim. This is another video on the paper 4.1. I've just completed one video on the question 1 and 2. Now we'll continue with 3 and 4. Starting with question 3, ecological surveys are conducted before conservation decisions are made. For example, surveys can be carried out before deciding whether to reintroduce a species to its former habitat. So once upon a time, maybe white lions were very common in a certain place, and now they've become extinct. So we said, okay, well, we've got a few more white lions somewhere else in the world, and we just bring 10 or 20 or 30 and put them back here in this former habitat. So decisions have to be taken whether we should reintroduce a species to its former habitat. Outline how an ecological survey can measure the biodiversity of a terrestrial habitat. Now, this whole question starts with that we're going to reintroduce, and then of course another part of it is questioned. Then it says table 3.1 lists some mammal species that became extinct in Great Britain or were introduced into Great Britain in the last 12,400 years. The reason for each extinction or introduction and the time of each event are shown. So, species Arctic fox, a number of years before present time, it was about 12,400 years ago, it became extinct because of climate change. Now the sheep 5,400 years ago were introduced for a farming. Mainly they were used by farmers and they were kept for slaughtering for the meat and all that. The house mouse was 3,500 years ago introduced, but was accidental. It wasn't something we did purposely, but it just happened accidentally. Maybe it came in some transport way, or maybe it was brought by somebody just as a pet, and then of course so accidental. Then the lynx 1500 years ago became extinct because people used to hunt them, maybe for their fur, maybe for their meat, maybe for some other thing which they had. Then fallow deer 900 years ago introduced as food. So this was used as food. Deer meat was used maybe to make kebabs or something like that, and was used as a food. Then the South American copu, and this of course 86 years ago, this was 33 years ago, introduced for their fur, but the other was introduced 33 years before the present time, and now they've become extinct. The reason is conservation culling. Now conservation culling, everybody should know the meaning of the word culling. Means when you purposely kill, or you actually decrease the number. They decrease the number of those animals in because of a certain reason. So that's called conservation culling. But you can look it up in the dictionary, and maybe then you can figure it out. Then the next part of the question is, with reference to table 3.1, state and explain the factors that have had a negative impact on the biodiversity in Great Britain. So basically you're going to pick up stuff from the previous table. You're going to pick up stuff from here, that how this has affected the biodiversity. So I'm reading the whole question to you, just making you develop the habit of reading these questions. Then part C, non-governmental and government organizations are working together to reintroduce a lynx population to its former habitat in a remote part of Great Britain. Lynx are predatory big cats. Okay, fine. So they've given you a picture of it. Suggest the factors that need to be considered by organizations to successfully reintroduce and restore a breeding population of lynx in Great Britain. Now you've got to think, okay, if you go to them, are there going to be enough food for them? Are they going to maybe result in, if their numbers increase too much, will they kill the sheep or the cows, or other farmers affect their livestock? So understand is that how they're going to be, what are the sort of questions which should come into your mind as a biology student. And then of course we go back to the question now. Now if you look at the mark scheme for the question 3A, now it was used random sampling. If you gave me the technique, or you just say randomly you choose numbers, and then you say, okay, you divide that whole area into random, and you say, okay, this is 1, 2, 3, 4, 5, and then you put all these numbers into a basket, and then you pick up randomly, okay, 4, 5, 7, 10, 11, what are the points where you are going to place what? You're going to place a quadrat, a frame quadrat, to measure the plants and the vegetation. or the stationary organisms. Then how do you detail using the quadrat? Okay, how are you going to place it? It's made of what? It's made of wood. It's 1 meter by 1 meter. It's got these threads in it which are dividing it into squares, and then you have to count the number of squares which has a certain vegetation, maybe white daisies, maybe red daisies, maybe yellow daisies. And then, so the first is use random sampling, then you use quadrats, and then you give me details of using the quadrat, that this is 1 meter by 1 meter, it's made of wood, and it's got these lines in it which are of course threads, just like a tennis racket or a squash racket. Then you repeat and find the mean. Well, that's probably you can write in any question. You can write it in lots and lots and lots of questions in which they ask you experimental procedures. Then if you're going to count the animals, then that's a mark, release, recapture. You take them, you set up traps for maybe rabbits or moles or some sort of small animals. They're not big animals. And then you catch them in this trap, and then in the next day you mark them. And then of course you release them, and then of course after 2 to 3 days then you capture them again, and then you see. So that's called the mark, release, recapture. Then detail of trapping, yeah, marking, or the named animals that you used. You gave me a little detail about that we'll take the rabbits, or we'll take moles, or we'll take some other animal, and then you mark them, and then you release them, and after 2-3 days then they mix, then you can of course recapture them. Then you calculate or use the Simpson's index of diversity. Now that's only you were writing it, even if you didn't know it, but you wrote it, that this is to calculate the Simpson's index diversity to get you the what is abundant, the species richness and the species evenness. So, and then of course the last point was species richness and abundance and species evenness. Now when it says here AVP, it means any valid point. So if there is anything which has not been mentioned here, but something which comes to your mind, and then of course the examiners will put it together and say, okay, well, this is worth a mark, and we'll give a mark for this. Now if you look at this table, that because in the next part of the question they had asked you is, what is this table mean? With reference to table 3.1, state and explain the factors that may have had a negative impact on biodiversity in Great Britain. So let's look at this table again, and let's look at the points which are available to us. So the first point which came to my mind was, okay, environment, climate change. So climate change was at the end of the ice age, or it resulted because of the local global warming which started occurring in place. So climate change, global warming was one mark. Then what happened to the Arctic fox? The Arctic fox was not so well adapted, so it sort of finished off. It extinct. Why did it become extinct? Because the climate change was so severe. Maybe the temperature rose, and this animal was just not adapted to that. So Arctic age, climate change because of the local global warming. So Arctic fox species were not well adapted. So that was one point. Then hunting, where is the hunting part? So hunting removed all the lynx. So hunting removed all the lynx, which are the large predators. Then introduction of new invasive species, like you introduced the fallow deer, which was for food, and the house mouse, which was accidentally. So introduction of these alien species, you named them, you said the house mouse or the fallow deer or the sheep, which was used in for farming. So you had introduced these new ones, and these could be competing with the other native species. So when you introduce them, these are the ones which you introduced, they could be competing with the native species. Now this is very important terminology, competing with native species. Native means the ones who are there beforehand. So competing with the native species, so this was another mark. Then may eat and spread disease to the native species. May, of course, if you introduce the deers could be eating, of course, all the grass and all that could be upsetting the balance as well. So may eat or compete with or spread disease to the native species. Then, of course, the problem of overgrazing, because there are too many herbivores. And this, of course, may change or destroy the habitat or certain food webs, because that would become not very viable, because certain animals would eat up all the, if they were all herbivores, then the grass would be less and the grasslands would not be enough. So then they would increase in numbers, but then they would start to die of starvation because there was no food. So referring to this table, you had to talk about different things which were relevant to this table. Then coming to this last part, which is they talked about this links, and it says suggest the factors that need to consider to successfully reintroduce and restore a breeding population. Now there's 5 marks. There's a lot of marks, and you have to be really very sensible in replying this. So number one, when you introduce new alien species, introduce healthy, fertile, captive bred animals. So you introduce healthy, wild, healthy, fertile, captive bred animals. Then because you would be introducing a few in number, so they must have a lot of genetic diversity or genetic variation. Then you should ensure that there's a sufficient habitat available for them. Then there should be sufficient food available for them, because if they eat grass, then is there enough grass? Do they eat other small prey? Then do they have those small animals available which they eat? Then sufficient prey or sufficient food available. And then public safety advice, then telling people that okay, well, if you see these animals, then you don't shoot them or you don't kill them, and they have been reintroduced into this habitat. They were there once upon a time, many hundreds of years ago. So sufficient sufficient food, and the fact that you give public safety advice and education to the people around who live around those areas. Then you also organize compensation for farmers who lose livestock. If their livestock is killed, if they are their sheep are killed by these lynx, then would you give money to them? Would you have enough money so the government would pay them the money? So compensation for farmers who lose livestock. Then outlaw the killing or disturbing the lynx populations which have been introduced. And some people might think, oh my god, they're dangerous, so let's kill them. So outlaw that, because you're introducing them back into the normal habitat. You want them to increase in numbers, to start adapting to live there. So outlaw killing them. And then monitor lynx and other animal prey populations. So you would monitor that. And the last thing was cull or use contraceptives to if the population grows too much. If the population is too much and now is bad, so then we call that conservation culling. So if population gets too much, then of course you need to then decrease them in number. Just a quick recap of this. Obtain healthy, fertile, wild-caught, captive-bred animals, genetic diversity, sufficient habitat, sufficient food, public safety advice, education, compensation for farmers who lose the livestock, outlaw killing of these, then monitor the lynx and the prey populations, and cull or control or use contraceptives if population grows too much. So 5 marks, but of course you had 9 points, and of course any valid point was also acceptable. Then coming to question number 4, in 1984, the geneticist Alec Jeffrey invented a DNA testing technique known as DNA profiling. Then that produces a DNA banding pattern on a gel. The DNA banding pattern is unique to each individual. DNA profiling can be used in police forensic work to catch criminals. In 1987, police in many countries have collected and stored DNA from crime scenes to create DNA profiles. Okay, so the police has record, okay, this person committed a robbery, this person committed a murder, so we've got his DNA. So let's make a DNA profile and keep it in a library and collected and stored DNA from crime scenes, which they try to match with the DNA profiles of criminal suspects. Okay, DNA at a crime scene may be obtained from hairs, traces of blood, semen, and saliva. Naturally, if there's a murder, then there's blood. If there's been a rape case, then there's semen. And of course, if there are glasses lying around or things where people have eaten food or drunk coffee or tea or something, then they've got the saliva on there, on the marks on those glasses and cups. So DNA at a crime scene may be obtained from hair, traces of blood, semen, and saliva. Explain why PCR may be needed before DNA from a crime scene can be profiled. Naturally, crime scene DNA is in very small amounts, and what does the PCR do? PCR amplifies the DNA, so makes millions and millions and millions of copies of it, so then that you can get an idea about the DNA, because you're going to do a number of tests on that. Explain why electrophoresis produces a DNA banding pattern. Naturally, what you do is you use restriction enzymes, you cut up the DNA into tiny fragments, and because it breaks up into, basically the main reason is the fragments break up into different sizes. If they were all the same size, then we wouldn't be able to do anything with the gel electrophoresis. But we have different fragment sizes. Like we have a fragment which is 100 base pairs long. So 100 base pairs long is, say, this big. So 100 base pairs means ATGC, and this is 1 to 100. And then there's one which is 500 base pairs long. So that's even longer, 500 base pairs long. And then there's one which is maybe 1,000 base pairs long. So there are different sizes of DNA fragments. So DNA moves to the positive electrode. Why? Because it's negatively charged DNA. It's got the phosphate on it, which is negatively charged, and this moves to the positive electrode. So please understand, and the reason is that the smaller fragments move faster, or you could have said the heavier fragments move slower, so they stay away. So say this is the gel electrophoresis, the gel, and you've got the wells here. So the smaller ones would of course go further away, the 50 and the 100 base pairs, but 1,000, 2,000 would be somewhere here next to the well. These khaddas or these depressions, these are called wells, where actually you put the DNA, where you put a comb and you left these little wells in it, wells. So these was where you're going to add the DNA. Then part B, GED match is described as an open data personal genomics website. It can be used by people who want to upload their DNA data to trace their ancestors and other elements. So these are people who are doing it willfully and who just want to find out who their ancestors or who their relatives, or maybe they lost some relatives, like when Pakistan India was made, so many people were left in India and some people got lost from their families. So if people want to find out where their families are, or maybe their ancestors or their relatives. In 2018, police in the USA solved a large number of serious crimes. Some of these crimes had been unsolved for over 30 years. So these criminals were roaming around and they were not caught or apprehended and not put behind bars. The police used GED match to profile DNA taken from crime scenes and to look for matching DNA profiles. In many cases, the police found partial matches to the relatives of criminals. So we found, okay, this criminal has a relative who lives in this town. So, well, maybe we can go there and ask him where his relatives are. This allowed the criminals to be identified and then charged on the basis of a complete DNA profile match. Suggested why the police strategy of comparing crime scene DNA with the GED match data was so successful. Why? Because database was very large. There were people who just wanted to find out about their ancestors and their relatives. Then police database only had crime suspects. So this was more, this was less. Then GED match was of innocent people who didn't really know actually that you were actually using their data for some other purposes. Now that's another ethical issue. Then provided multiple leads to guilty people who are hiding their DNA. So this is how it helped a lot. Then the next question which they've asked you is, explain why GED match an example of bioinformatics. Why? Because it's a lot of biological data. It's nothing else, but it's a lot of biological data. What was bioinformatics? It was all about biological data being stored in websites. So biological data, then for processing, for analyzing, or for processing, we wanted to analyze it and we wanted to really figure it out what gene is present in which organisms, and by computer softwares. So if you knew bioinformatics, if you knew the word, of course in the new syllabus the word is not being used, but the bioinformatics is not being used. Now they've just said that you have to know how all this database is maintained and all that. Then coming to question number 3, the first successful conviction resulting from the use of GED match by the police was widely reported. Some journalists and broadcasters thought that the GED match website should not have been used by the police in this way. In the days following the news, the number of citizens choosing to upload their DNA data to GED match increased from 1,500 to 5,000 a day. Comment on the social and ethical issues raised by the first successful conviction. And that's for 2 marks, and this is suggest, so it's a little tricky. So of course this helps the police to catch criminals. So if the police is effective in catching the criminals, so it's a good thing But whenever they ask you such a question, is social and ethical issue, so write a few favorable points and write two which go against it. Make society safer. Why? Because if the bad guys are behind bars, then we have less thefts, less crimes, less stealing, less rape, less shoplifting. So we have less problems. So make society a safer place. Then, of course, DNA breakthrough helps justice and law and order. Helps law and order. So it helps the police in maintaining law and order. But sharing own data and sharing relatives' data without their consent, that was another thing. So you were sharing data without their consent. Now that was an ethical issue because you hadn't said that to them, you hadn't told this to them. So sharing own data also shares relations data without their consent. So you had to share your DNA, but then they had information to your relatives' DNA because some of the DNA matched with that. So sharing own data also shares relations data without their consent. Question 5a. Gibberellin is a plant growth regulator involved in barley seed germination. Production of gibberellin is stimulated by the uptake of water. State the location of gibberellin synthesis in a barley seed during germination. That was embryo. Then barley seed germinates when placed on blotting paper soaked in water. The germination of barley seeds placed on blotting paper soaked in solutions of different water potential was investigated. The success of germination was measured as a germination index for barley seeds placed on boiling paper soaked in water, and for barley seeds placed on blotting paper soaked in 5 solutions of different water potential. The results are shown in figure 5.1. The higher the germination index, the more successful the germination of the barley seed. So anything which was 50% germination index, it meant 50 was a higher rate than as compared to 10 if the germination index was 10. Now with reference to figure 5.1, describe the relationship between the germination index of barley seeds and water potential. Now if you look at the water potential, this is 0, so this is the highest. And as we are adding solutes, minus 0.5, minus 1, minus 1 point, minus 2, minus, so it is decreasing in this. So the water potential, as I told you, 0 has the highest water potential. So if it's distal water, highest water potential is distal water. So that is 0.0. Now with reference to figure 5.1, describe the relationship between the germination index of, so it's pretty easy. You can see it's generally decreasing. And then if you gave me 2 figures, well, that's how you got the 2 marks for it. There are only 2 marks for that. So the basic whole story from the graph was, as the water potential increases, the germination index also increases. You can see here the water potential is decreasing in this direction, but it would be increasing in this direction. So as you can see here, the germination index is less, and here the germination index is more. So as this water potential is increasing this way, 0 is the highest water potential, or you could have got it the other way around. As the water potential is decreasing, the germination index is decreasing. So as here it says the water potential, you can say anything, both of this was allowed. So reverse was allowed as well. So low water potential decreases the water uptake, water needed to activate embryo, which embryo produces gibberellin, then water used for hydrolysis, then starch is converted to maltose, maltose to glucose, water is a medium for reactions, and lowering the water potential may inhibit the enzymes. So these were the explanations that you had to give me. Then it says during barley seed germination, gibberellin stimulates synthesis of the enzymes. Still state the name of one of these enzymes. Or you could have said amylase. You could have said maltase or protease. Any of these was correct. So any of these you could have said, because that's what is going to be released when the germination starts. And then the location of this was, of course, very simple. It was the alluron layer. This, of course, mentioned in the book as well, and it's something which we all know about. Then it says some plants are grown commercially for their flowers. Many of these plants are varieties that have short stems. Two factors that affect the height of stems are gibberellin and the capital L small le gene. The capital L small l gene has two alleles. I'm not talking about the E part. So there is a capital L. So this is the homozygous dominant. Then there's a capital L and then there's a small l. And then they're both small l's. So homozygous, heterozygous, and homozygous recessive suggest an advantage of growing a short-stemmed variety of a flowering plant. Then of course part 1 suggests an advantage of growing short-stemmed. So flowers will be, if you have short-stemmed, these plants are not going to topple down by wind or heavy rain or storm. So flowers less likely to be damaged. Then it says explain how the capital L and small l gene and gibberellin are involved in affecting the height of plant stems. So coming to the last explanation, the L gene and gibberellin are involved in affecting the height. So short plants will always be homozygous recessive. Now that was something which you gathered from the question. Recessive allele codes for the non-functional enzyme. So non-functional enzyme means inactive gibberellin produced, DELA protein not broken down, PIF cannot bind to start transcription of genes that promote cell growth and cell division. You see, basically tall stems are a result of number 1 mitosis, cell division, then of course it is cell elongation, and then cell differentiation. So first it's cell division, elongation, and then differentiation. So that finishes this video, and we will continue the questions left in the next video. And thank you very much once again.